Welcome to the third screencast in Chapter 11, dealing with the molecule DNA and how we eventually get to the expression of traits. So you're all experts in DNA structure, the components of DNA, the shape and the structure of the molecule, and what the bases are in DNA and how that actually works as a code for eventually a particular trait. But just remember, so you start with a DNA molecule, and a particular segment of a DNA molecule, a specific sequence of bases, is a gene. And genes are eventually going to code for proteins, and proteins are probably the biggest class of molecules in living things, and eventually a protein is what's actually going to determine a structure in the human body or a chemical in the human body that basically describes the function or the structure of a part of the human body. So this slide is looking at, again, a big picture of this. Remember that DNA, the molecule, is always found in the nucleus of a cell. Well, proteins, the proteins that are eventually going to carry out a trait, whether it's eye color, skin color, or hair structure, or blood type, etc., proteins are actually manufactured out in the cytoplasm at the ribosome. So we really kind of need a middleman. We need something to take the code from DNA and bring it out to build the protein. Well, that middleman is RNA. So remember this sequence that DNA makes RNA and RNA is going to make protein. This picture shows you transcription. This is this RNA. We'll talk more about its structure and what it is in a second, but this is RNA, which is made specifically off the basis of a DNA molecule. There is nothing random about the process. So first of all, we need to answer the question of what is RNA? What is ribonucleic acid? And there's some similarities between DNA and RNA, and there's some three very important differences. RNA, unlike DNA, is a single strand. So here's a model of an RNA molecule versus a DNA molecule. It also has a different sugar, and there's one difference in the bases. In RNA, the base uracil takes place of the base thymine. So in RNA there is no thymine, and in DNA there is no uracil. For comparison, you take a look at the DNA molecule versus an RNA molecule, and again these differences are pointed out. So there's actually three different types of RNA. There's an mRNA. The first type is an mRNA called messenger RNA. The M stands for messenger. And this is really the big player in what we're talking about today with making RNA. We're going to use mRNA as our example. But again, RNA is made directly complementary to the DNA. As you can see, C goes with G. U is going to go with A. Remember, there is no thymine in RNA. A goes with the T on DNA. So these bases are complementary to the bases in the DNA. That way, everything is very specific. The second type of RNA is called transfer RNA, or tRNA. As you can see, the actual model or picture of this roughly looks like a T, the letter T, but it is a transfer RNA. Its main job, which we'll get to more of later, is that it has a specific anticodon. We'll get to that term later, but these three bases are going to match up with mRNA. Again, complementary speaking. But the main job for now is that they transfer amino acids. Remember that amino acids are the building blocks of proteins. Our RNA, the third type, ribosomal RNA, actually makes up the cell part called a ribosome. Transcription, the process, we keep it to three pretty simple steps. More is going on here, but much like replication, the DNA molecule needs to unwind and unzip so that it exposes these bases. But as you can see in the picture here, is that as it has exposed bases on DNA acts as a template, and the growing RNA strand is going to be built complementary to one side of the DNA molecule. Then, once this is completed, the RNA sequence or the RNA strand is able to leave the nucleus, go out to the ribosome where proteins can actually be put together. So if you were viewing this as an actual PowerPoint, this would be an animation, and these different strands we'd be able to maneuver them. But if you take a look at this, you see this piece right here represents a DNA molecule. It's double-stranded, and of course we're just labeling the important parts, which for us are the letters, which represent the bases. And this is a single strand of RNA. Now what you should be able to do is you should be able to see which strand of the DNA molecule was used to make that RNA. Did we use the one on the left or the one on the right? In other words, which DNA strand is this RNA complementary to? Of course, looking at it, you should note that the U would be complementary to the A, C complementary to the G, G complementary to the C. So in other words, we used the strand on the left of DNA to transcribe or make this mRNA sequence. So again, you should be able to go back and review this screencast, back up this PowerPoint and screencast, and be able to slow down and look at those different steps of what's actually happening with the DNA to RNA transcription process. Remember that the DNA never leaves the nucleus, so transcription is the process of making the RNA molecule. There's nothing random about it. These bases in this RNA sequence are very specific to the original code, and that's in turn going to give us specific proteins and specific traits that make us individually who we are, but even broader than that, makes all living things with the genes, the collective genes and traits that all living things have, what makes them unique.